Good morning, everyone, or good afternoon, wherever you are in the world. Thank you for joining us for this sixth webinar offered by the group Polyphenols. The group Polyphenols Society is an international association founded in France in 1972 with the aim of promoting research on plant polyphenols from their most basic and fundamental biophysical-chemical properties to their most diverse applications in food and agricultural, pharmaceutical, and cosmetic sciences and technologies. The group Polyphenols started to develop this series of webinars on Polyphenols Research in 2022 with presentations about polyphenols in soil systems, food and beverages, and about their characterization. If you didn't have the chance to participate in the previous webinars, you can have access to the recordings on the website of the group Polyphenols. I'm Dr. Aude Watrelot, an assistant professor and extension specialist of enology at Iowa State University, and I'm focusing my research on polyphenols and their chemical properties in wines. I'm the vice president of the group Polyphenols, and I'm co-hosting the webinar today with Dr. Nuno Mateus. Before I give the floor to Dr. Mateus, I would like to provide you some instruction about the webinar. So the webinar is currently recorded, and it will be available on YouTube on the group Polyphenol website in about a month. Please type your questions in the chat box that you can find at the bottom of your screen by a click on the three dots. Your questions will be answered at the end of the webinar. Also, at the end of the webinar, you're going to receive an email to complete a Qualtrics survey about how we did today and what we would need to change for future webinars. So please take a few minutes to complete the survey. Also, if you need a certificate of participation to this webinar, please send me an email to request the certificate. You will then receive it within a month. So now it's my pleasure to give the floor to Dr. Mateus, who is going to introduce himself and the speakers of today. So Nuno.
Thank you, Aude. Good afternoon, everyone. So my name is Nuno Mateus. I am a professor at the Faculty of Science of the University of Porto in Portugal. And I've been working with polyphenols since over 30 years, and especially in wine chemistry. It is my pleasure to host this sixth webinar in Polyphenols Research. I'm a member of the group Polyphenols, and the theme of this webinar is Polyphenols, Nutrition and Health. We have two speakers. We will begin with Dr. Ricardo Dias, and it is my pleasure to introduce Dr. Ricardo Dias, since Ricardo works in our group here at the University of Porto. He will deliver a presentation titled From Food to Health, the Role of Polyphenols in Modulating Celiac Disease Pathways. So Ricardo Dias graduated in biochemistry in 2013 and obtained his biochemistry master's degree in 2015, both at the University of Porto in Portugal. In 2020, he got his PhD in sustainable chemistry. His field of research concerns molecular nutrition and food science, with particular interest in understanding the chemical and biochemical pathways by which polyphenols modulate the onset of celiac disease. By taking advantage of the antioxidant, anti-inflammatory, and protein-binding properties of polyphenols to downregulate both innate and adaptive immune responses to gluten, proteins, and peptides, Ricardo Dias aims to open new horizons concerning the rational development, applicability, and valorization of polyphenol-rich foods and industrial byproducts as promising nutritional complements to a gluten-free diet. Dr. Ricardo Dias, it is my pleasure to welcome you, and the screen is yours now.
Good afternoon, everyone. First of all, I would like to acknowledge Professor Aude, Professor Nuno for the kind introduction, as well as Polyphenols for inviting me to discuss From Food to Health, the role of polyphenols in modulating celiac disease pathways. A topic that not only highlights a compelling intersection between food science and human health, but also delves into the potential of polyphenols to impact celiac disease management. So we'll begin by looking at the current trends in human nutrition, setting the stage for a closer look at how polyphenols uniquely fit into this context and the promising doors they might open concerning the improvement of the quality of life of people affected by this condition. So, as you all probably noticed already, the food shopping experience is nowadays as dynamic as ever, and from the rise of non-traditional food sources to new attitudes about health and wellness, things have definitely changed because today consumers are looking for customizable foods, beverages, and dietary solutions that can help them to more aggressively meet their own unique nutrition and personal health goals, for which high protein, vegetable-rich foods or superfoods are the most attractive to dinners, while heart health, weight management, energy, or gut health are some of the most influential issues. But there are more, and there is one in particular that has been driving considerable efforts by food business operators when conceiving and developing novel functional food products. That is the rapid spread, the rise in food intolerance and food allergies to dietary proteins, and including from plants like wheat, that although is not life-threatening as other non-communicable diseases, they can be as distressing as those. And this graph illustrates the serious disease, in this case for celiac disease, whose incidence has been considerably increasing over time among all countries and ages, and to which the only available treatment so far consists on a lifelong adherence to a gluten-free diet. And even though for most celiac disease people, adhering to a gluten-free diet is effective in stopping symptoms, healing existing intestinal damage, and preventing further complications, for others, however, gluten withdrawal from the diet is simply not enough. They still develop symptoms despite their best effort to avoid gluten. Also associated with gluten-free diet are a series of nutritional, socioeconomic, and psychological challenges, not to mention how hard it is for these people to avoid inadvertent gluten ingestion, either because of misinterpretation of food labels or cross-contamination of food products. So these are all common struggles these people must face day in and day out throughout their life, especially teenagers who often consider themselves as outsiders or a burden on others because of their different diet. Generally speaking, and without going into too much detail, celiac disease is a chronic immune-mediated enteropathy that is triggered by dietary gluten, which is present in cereal grains such as wheat, barley, rye, and some oat varieties. And the pathophysiological mechanisms of celiac disease, as you might all see, are highly complex, some not yet unknown, but still they all depend on the expression of two very specific HLA class II genes, known as HLA-DQ2 and HLA-DQ8, which are responsible for binding and presenting completely digested gluten peptides to gluten-specific T cells, which in turn will license intraepithelial cytotoxic lymphocytes to kill activated epithelial cells based on the recognition of stress signals that are induced by gluten itself. And the results will be like this. I mean, an architecturally diseased intestinal mucosa is histologically characterized by villous atrophy, enlarged hypoplastic crypts, and higher infiltration of intraepithelial lymphocytes. As the focus on celiac disease has intensified in recent years, so too some concerns have been rising because several studies now suggest that celiac patients' guts are unlikely to heal completely on such a diet, and that the accidental ingestion of gluten is almost inevitable, essentially because gluten is virtually ubiquitous in today's world, found in just about everything we eat and drink. So new solutions are needed, and that's exactly through this window of opportunity by which natural products are becoming once again increasingly influential in food service. Natural products that actually and historically speaking have always played a key role in drug discovery and drug development, especially for cancer or other infectious diseases, but also in other therapeutic areas such as cardiovascular diseases or even to multiple sclerosis, for instance. But presently, and mainly because of these behavioral twists seen in recent years regarding the growing consumer interest in innovative, sustainable, and plant-based food solutions, delivering both mental and well-being benefits like immunity and stress management, the interest in natural bioactive compounds is being revitalized, among which polyphenols stand out and are being truly researched for their health-promoting bioactivities in many disease contexts, including celiac disease. But the question is, why polyphenols? Why should we bother with polyphenols? And putting into perspective, these outbreaking polyphenol-oriented researchers can perhaps find justification in an important property that alphenols share to some extent. That is their ability to scavenge oxygen and nitrogen-free radicals, thereby forming the aroxyl radicals. And so this is important for disrupting oxidative chain reactions that may damage cellular compartments and including proteins, nucleic acid, or even the plasmatic membrane. So it's like they prevent oxidative stress caused by these species. And actually, this antioxidant activity of polyphenols has been frequently cited to be the main event responsible for the prevention of age-related diseases, including neurodegeneration, carcinogenesis, or cardiovascular diseases. When people are consuming these polyphenol-rich diets. And such, as a consequence of such a potential for preventive therapies, antioxidation has become like the trademark for and the fundamental basis behind the use of these polyphenol substances, phenolic substances, in both for cosmetic or pharmaceutical products. We need to be cautious, however, because some of these aroxyl radicals that are formed by homolytic hydrogen atom abstraction or by one electron oxidative quenching of free radicals can be further converted into highly electrophilic non-derivatives, which are highly toxic if they escape the redox cycle. So here we have the same way they are antioxidant compounds, they can also become peroxidant depending on the including from the medium, for instance, on the pH is an important factor that modulates this balance. Beyond these chemical, these bioactivities, which is based on a chemical reactivity that is inherent to the phenolic function, it has also been shown that polyphenols can also specifically and physically interact with biomolecules and modulate cell signaling pathways that can modulate both cell death and survival under both normal and pathological conditions. And this is just a scheme to illustrate the different targets which have been already identified as being modulated by polyphenols and their relationship along with several non-communicable diseases. So getting back to celiac disease, as I previously mentioned, most of celiac disease immune reaction start once these immunoreactive gluten peptides get into contact with intestinal mucosa, where they trigger an impairment of the epithelial tight junctions, promote an activation of gluten-specific T cells in the basal compartment, and then cause a mucosal inflammation. But what would happen if we, besides the antioxidant and anti-inflammatory properties of polyphenols, we took advantage of the protein and peptide binding properties of polyphenols to neutralize or at least reduce the interluminal availability of immunoreactive gluten peptides. Because if we were successful, this would mean avoiding all the downstream pathways that perpetuate intestinal damage by either blocking epitopes or binding sites that are capable of being recognized by these HLA presenting molecules. So that was exactly the pathway I followed during the first years of my PhD, where I studied the interaction between different phenolics, mainly tannins, because they are well known for their protein and peptide binding properties, with one of the most bioactive gluten peptides, the 3233-mer peptide, whose sequence is here and is often considered to be the superantigen of celiac disease because it can trigger an adaptive response in most of the patients identified so far and has a very low threshold for recognition. So I started characterizing these interactions by nuclear magnetic resonance, and I did this by titrating the increasingly higher concentrations of epigallocatechin-galate. That's the polyphenol I started with. Most of you are probably familiar with epigallocatechin-galate as one of the main green tea phenolics. And what we observed is that the titration concentration increments of epigallocatechin-galate induced a progressive broadening, an intensity decrease, and upfield chemical shift perturbations in most of the peptide resonance signals, indicating the occurrence of an aromatic shielding effect because of the interaction between them. And actually, the backbone amide proteins were significantly more affected than most other signals, including the aromatic or aliphatic signals, which actually reflects a higher sensitivity to the surrounding chemical environment, and particularly on the establishment of polymer interactions with the solvent media or with specific functions of epigallocatechin-galate that has been added at the titration point. So by following these chemical shift perturbations induced on a very specific proton resonance that we identified as being a leucine residue on the 32-mer peptide, we were able to represent the magnitude of this chemical shift perturbation per micromole of added tannin. And this is a tannin I tested at that time, catechin, the porcine dimer B3, porcine trimer C2, epigallocatechin, and epigallocatechin-galate. And what you observed is that when the tannin polymerization increased from a catechin to a porcine trimer C2, or even upon granulation, these structural changes lead to a significant proportional variation in these chemical shift perturbations, indicating slightly different binding affinities, which are actually known to be favored by these tannin structural features. We also followed the 32-mer peptide changes into 32-mer peptide solubility by nuclear magnetic resonance that determine the percentage of peptide that remained in solution upon concentration increments of these several polyphenols. And again, as you can see, these trends we observed were different depending on the polyphenol. And again, while epigallocatechinylate and the trimer were found to be the most reactive polyphenols against the 32-mer peptide, and in the low concentration window for prosinidine B3, for instance, changes in the peptide solubility only started to be noticed at 1 millimolar concentration. And then from this concentration onwards, the behavior was similar to that of the trimer and the epigallocatechinylate. Well, even though these chemical shift perturbations we observed by NMR could not be assigned to specific amino acid residues, and this was because the 32-mer peptide, as I showed previously on the sequence, it consists of at least 3 motifs that are repeated along its sequence. So it's very difficult to define, okay, this glutamine is here or is it in the second or the third motif. So it was these resonance sites, most of them in the amide region, could not be assigned to specific amino acid residues, but rather the class of amino acids. And by following, and what we observe by following these chemical shift perturbations on the amide region, we observe that the most significant chemical shift perturbations involve both leucine and tyrosine amino acid residues, with changes in the characteristic frequencies starting to be noticed as substoichiometric amounts of ligand, and which suggests that these 2 amino acid residues may constitute key elements to which polyphenols will preferentially bind. And this was actually subsequently confirmed by using a bioinformatic tool, that is the Proto scale tool from Expazy, which allows the user to map the presence of hydrophobic motifs within a peptide sequence or a protein sequence. And this allowed us to see that actually both tyrosine, leucine, and also the phenylalanine residues were located into 4 almost indistinguishable hydrophobic clusters that were equally spaced by non-polar proline residues. So these are most likely the primary binding sites to which a polyphenols will initially bind with more or less affinity depending on their nature, like the structural complementarity with the peptide, for instance. We then move to STD-NMR, which is one of the most popular ligand-based NMR approaches for studying protein ligand interactions. And it relies on the fact that for a weak binding ligand, there is an exchange between a bound and a free ligand state, to which a protein or peptide saturation is selectively transferred to the ligand by a nuclear Overhauser effect. And in practice, for a molecule that binds to a receptor, in this case, we consider the receptor as being the 32-mer peptide, only the proton signals that are contained with the moieties that are closer with the peptide will receive this magnetization transfer and then appear in the different spectrum when we do these kind of experiments. And this allows us then to determine, okay, for these 3 ligands, the epigallocatechinylate, the trimer B3, and the trimer C2, which are the structural moieties that are closer to the peptide that's closer, I mean, like on radius of less than 5 angstroms, which in epigallocatechinylate was found to be the Galil groups, while for the dimer and the trimer, it depended on the unit. Of course, we couldn't finish these interactions without performing isothermal titration calorimetry, which allowed us, and we did this at 2 different temperatures, 25 and 37 degrees Celsius, and allowed us to determine a series of physical, chemical, and thermodynamic parameters, including association constants, binding stoichiometries, enthalpy of bindings, free energy of Gibbs, and entropy changes associated with these interactions, which were found to be multiphasic. This is for prosinidine B3, and actually the iteration is quite simple. It's like a 1-to-1 interaction, while for prosinidine C2 and the epigallocatechinylate, it was found to be multiphasic. So it cannot be described as a simple 1-to-1 model, binding model. And what was observed is that the interaction between the 32-mer peptide and all these polyphenols were found to be spontaneous at both temperatures, and entropy driven, because the enthalpy contribution on the free energy of Gibbs is residual when compared to the entropy contribution. So according to this result, it is expected then that the hydrophobic interactions should drive the binding process between the peptide and these polyphenols due to the isolation entropy and water release from both the aliphatic and the aromatic side chains of both leucine, tyrosine, and also phenylalanine residues. and that the establishment of hydrogen bonds will then strengthen and stabilize these format complexes. So these are actually in line with observations we got from by nuclear magnetic resonance, and later we also performed some molecular dynamic simulations which also confirm these trends. So once we characterize the interaction between the 32-mer peptide and epigallocatechin at the beginning, and we characterize it by both molecular and thermodynamically, we performed some in vitro transwell experiments on cacochu cells that were differentiated to see whether epigallocatechin was able to neutralize the transport of the 32-mer peptide by complexation. So what we did, we prepared a mixture of the epigallocatechin and the trimer and the peptide, and we applied on the apical compartment of these transwell systems where cacochu cells were allowed to grow and differentiate for 21 days. And then we took, at the end of 4 hours, we took basal liquids from a basal-lateral chamber, which we then processed and analyzed by mass spectrometry to quantify the amount of peptide that got able to translocate to this epithelial barrier. And what we observed is that in the absence of epigallocatechin, around 2.5% of the peptide was able to cross from the apical to the basal-lateral compartment, whereas in the presence of epigallocatechin, the peptide was almost not detected or when it was present, it was basically in the background noise level. So these results were interesting because they clearly provide some clues that actually polyphenols, by complexing these immunoreactive peptides, can actually modulate the translocation. And of course, this has a lot of implications for instance, reduce, attenuate the subsequent cellular and molecular mechanism that perpetuate celiac disease, like activation and triggering of gluten-specific T cells and later mucosal inflammation. But at this point, we question ourselves: okay, if in conditions where we have a strong complexation between the peptide, the 32-mer peptide, and epigallocatechin with such a remarkable attenuation of peptide transport, what would happen if instead we use substoichiometric amounts of polyphenols, and preferentially in conditions where their peptide binding properties and supramolecular effects could be neglected? And that's what we did. First, we reassessed the ability of both epigallocatechin, the trimer C2 and the dimer B3 to interact with the peptide. And we did this by fluorescence quenching. I do not present the results here, but just to get an idea whether at substoichiometric amounts of ligand, we were able to see what would be the trend in their binding association to the peptide. And what we observed is actually that the B3 and the trimer C2 had like similar association constants at physiological conditions. That was an order of magnitude lower than that of epigallocatechin. So for these experiments, transwell experiments, rather than use the epigallocatechin, we assess the ability of substoichiometric amounts of B3 and the trimer C2 to modulate this peptide transport in vitro. And this is where things become interesting, because even though both the dimer B3 and the trimer C2 had similar association constants to the peptide, they were both able to modulate and to reduce the translocation of the peptide in vitro in a trend that appeared actually to be dependent on their polymerization degree. And later we also observed that even the way the cells perceive this peptide and metabolize them, and by metabolizing I mean like this peptide gets cleaved both at the apical surface of the enterocytes or even the transcytosis process, the way it was metabolized and processed was significantly different. So for sure, there are other mechanisms that are in play here. We are still working and trying to dissect them here, but these results are very interesting and very promising in the sense that depending on the polyphenol, both for sure the epigallocatechin trend is similar at substoichiometric amount of ligand, but even without a strong complexation between the phenolic and the peptide, these polyphenols are able to modulate the transport in vitro. And this could be explained, for instance, by the upregulations, for instance, of the tight junction proteins, which are actually known, and several studies now show that in different disease contexts, that they are able to increase the expression of, for instance, occludins, claudins, or some occludins. So, okay, at this point, all these results were very nice, very interesting, but we wanted to go a little bit further, and because most of these results were in vitro, so we wanted to see what would be the effect of polyphenols in celiac disease relevant in vivo models. So for this reason, in the last part of my PhD, I moved to Italy, where I had the chance to access the efficiency of epigallocatechinolate and two polyphenol-rich extracts. One that was prepared from Portuguese green tea, which actually has epigallocatechinolate as its main constituent, and another one prepared from grape seeds, so it's basically composed of oligomeric procyanidins. And I tested the efficiency of these extracts and epigallocatechinol on a decubitogenic mouse model of celiac disease enteropathy. And this is like the study design I followed at that time. So at the beginning, we had 25 animals, which I divided into 5 different groups. One group was left untreated and served as a negative control, while other 3 groups received by oral route around 50 milligrams a kilogram a day of both epigallocatechinolate or the catechins extract or the procyanidins extract, 2 weeks before being intragastically administered with 25 milligrams a kilogram a day of gliadin proteins that was given every 2 days. And the application of polyphenols continued during this time period after which animals were sacrificed and analyzed by biochemical and microscopy. To generate enteropathy, and this was mandatory for this model because this mice model didn't develop enteropathy without only by gliadins. They had to have a co-stimulus, and this was indomethacin that was given in the drinking water. And what we found out was that, as you can see from this figure, at the end of 30 days, decubate mouse received only gliadins and subenteropathic doses of indomethacin showed a significant reduction in the villus high, increased grip depth, and a significant reduction in the villus high to grip depth ratios. And what was interesting is that these structural changes were somehow attenuated by polyphenols intake. Even though for some parameters we couldn't reach some statistical significance, but here when we look at the values high to grip depth ratios, we can clearly see that there is a protective trend played by these polyphenols. So it does appear that polyphenols tend to counteract the toxic effect of gliadins by partially restoring this homeostatic equilibrium between cell apoptosis, cell proliferation at the grip level, and cell apoptosis at the top level of the villus. Features that are essential for the polyphenols for the intestine to maintain its integrity is absorptive capacity and growth efficiency. Also, what was interesting to see is that the regular intake of green tea catechins, particularly green tea catechins, was highly effective in reducing the number of intraepithelial lymphocytes. So this is probably the consequence of an effective protection of the intestinal mucosa to a basal homeostatic state that ended up being less vulnerable to the immunostimulatory effects of gliadin. So all this data provides valuable in vivo evidence regarding the protective effect of polyphenols at Dietary as natural molecules that are capable of delaying some of the most structural abnormalities found in celiac disease. And this then we accessed also the nucleophilic tone at the intestinal level of these mice, and what we observed is actually the polyphenols favor the maintenance of intestinal redox homeostasis by significantly boosting the levels of total glutathione. Okay, lastly, what about the effect on T cells? We couldn't have really studied the adaptive range on these mice because they are not a model for that. They are just a model for the first steps on celiac disease onset. These are morphological changes that are triggered by gliadin. So we performed some in vitro experiments where we basically isolated and propagated gluten-specific T cells from intestinal biopsies of celiac disease patients. And we analyzed then the ability of green tea catechins to reduce the activation of gluten-specific T cells by gliadin peptides. And here is the results we obtained on PBMCs, which we observed like as a measure for the effects of polyphenols on a non-specific activation of gluten-specific T cells, and we observed that actually these green tea catechins are able to modulate both cell proliferation and cell activation markers on PBMCs. But the most interesting thing was to observe that they can actually reduce the production of interferon gamma, which is the main mediator on the adaptive range of celiac disease, in a dose-dependent way. So at 100 micrograms milliliter, we observed is more significant, but actually you observe by flow cytometry that the viability was also compromised on these cells. But in any case, there is a trend here that is very interesting to see, that beyond the morphometic and or innate branch of celiac disease, polyphenols can also modulate this adaptive branch. I would like just to end with a short introduction to a project that is currently ongoing here, and which we are studying making like a risk-benefit assessment of pigmented wheat varieties that are being developed in the partnership with the Czech Republic Institute in Czech Republic, and these wheats are basically characterized by increased levels of phytochemicals, including polyphenols, but also carotenoids. So we are using an integrative approach and multiomic approach to assess whether by having a matrix in which these gluten and polyphenols coexist, what would be the effect on both metabolization pathways and the effect on the celiac disease models. And we instead of using the in vivo model, we are now trying to develop the organoids model. So we have a lot of results right now. I'm not presenting them here, but stay tuned for the development on this topic. Yeah, and I would like to acknowledge to my supervisor, Vitor, to Nuno Mateus also, and all my colleagues from the Polyphenol Lab for their support. And yeah, and if you have questions, wait until the end of Jess' presentation. Thank you very much.
Thank you very much, Dr. Ricardo, for this very nice and very clear presentation. And so to remind everyone, we will leave any question for the end of the second presentation. So without any further ado, it is now my pleasure to introduce our second speaker of this webinar, Dr. Jess Reed. Dr. Jess Reed is an emeritus professor of nutrition at the University of Wisconsin-Madison with 40 years of postgraduate experience in research on the effects of dietary phytochemicals on the nutrition and health of animals and humans. He also leads the Reed Research Group, which consists of 3 core programs: phytochemistry, cardiovascular, and mucosal immunity. Extensive research collaboration has been developed across all 3 cores. The main thrust of the phytochemistry research core is to determine the effects of oligomeric polyphenols, also known as tannins, in foods on disease processes. This research effort includes development of phytochemical methods for characterization of structure and their interactions with proteins and polysaccharides, and mechanistic studies on the effects of tannin in cell culture and animal models of disease. The research core develops methods for isolating tannins from cranberries, sorghum, grapes, pomegranates, and several other food and forage plants in order to determine structural features by modern methods of mass spectrometry. They are currently studying the relationship between tannin structure and the ability of tannins to inhibit the adherence, invasion, and colonization of mucosal epithelial cells by extraintestinal pathogenic E. coli and other bacteria that infect the gastrointestinal tract, the urogenital tract, and also mammary glands. And last but not least, Dr. Jess Reed was the organizer of the previous 2018 ICP, the 29th International Conference on Polyphenols. So it is my pleasure to introduce Jess, and the title of his presentation today is Challenges to Research on the Effects of Dietary Polyphenols on Atherosclerosis and Cardiovascular Disease. Dr. Jess Reed, the screen is yours.
Let's see if I'm going to be able to advance my slides now. There we go. Atherosclerosis is a serious disease, and it's the main cause of a more generalized disease called cardiovascular disease, which is the disease of the arteries that lead to both heart attacks and stroke. And cardiovascular disease in the developed world is of epidemic status, because in the world, 32% of all deaths is attributable to cardiovascular disease, according to the World Health Organization. And in the United States, over 50% of all deaths in the United States are caused by cardiovascular disease. And that is a tremendous health cost of over $400 billion a year. So this disease is a serious disease. And this is what it looks like. This is what it looks like actually in a pig model of the human disease, but this pig model actually develops atherosclerosis very similar to humans. And so this is a normal coronary artery. This is an artery that carries blood to the heart muscle and is absolutely essential for normal heart function. And so you can see that there's an endothelium here, and the lumen is completely free. And these other layers are a layer of the media and the adventitia. So we have the endothelium and intima, and they're not thickened. In the extreme case of atherosclerosis, in the situation where the artery is completely occluded, the blood flow to the downstream tissue is completely blocked off. And when this narrow opening here gets hit by a thrombus or a blood clot, it causes a myocardial infarction in which the heart actually stops. And that's the biggest cause of heart attacks. Now, if this happens in the brain, then you're blocking off blood flow to sections of the brain, and that's the biggest cause of stroke. So it's a serious disease, and treatments for it are actually relatively poor. In fact, the most important preventative action that physicians can take are prescribing statin to try to lower blood cholesterol, and or putting a stent into the artery where these occlusions occur in order to allow blood flow to the peripheral tissue, our downstream tissue. Both of those are moderately effective, but they're not curing the disease. They're just treating or preventing death from the disease. And so what's fascinating about this development is, in fact, the incidence of atherosclerosis is probably still going up in the United States, but death from heart attacks and strokes are going down because of the rapid response to the problem in individuals, and also the intervention of having the stents placed. So what is the basic cause of atherosclerosis? It's fascinating, and it's an interesting disease because it happens over the lifetime of normal individuals, and it's thought to occur as in two processes. The first one that I'm talking about here is an inside-out process in which the endothelium is activated and attracts monocytes, and that is thought to be a response to oxidized low-density lipoprotein in circulation. And the accumulation of oxidized low-density lipoprotein in macrophages in the intima, which is this area here shown in the diagram, is in fact the beginning process. And those at early stages are referred to as foam cells to form fatty streaks on the artery wall. But the accumulation of foam cells and the oxidative processes that occur in the macrophages leads to the chronic inflammatory response that leads to the advanced lesion, as shown in the previous slide. Now, that's the inside-out approach, but in fact, in more recent times, there's also a new understanding of the development of the disease, which involves the injury, stress, and inflammation causing neovascularization, that is the formation of new blood vessels in the wall of the artery. And because the large arteries of the body require their own circulation and arteries and capillaries, that is referred to as the vasa vasorum. And it's thought now that macrophage T and B cells are recruited to the adventitia, media, and intimal layers through the vasa vasorum. And it's the accumulation in lipids and inflammatory cells that lead to the propagation of the advanced lesions. So in fact, in both cases, we're talking about a chronic inflammatory response that leads to advanced lesions. So what are the risk factors for atherosclerosis and cardiovascular disease? Well, it's fascinating among, when you look at the epidemiological studies that look at risk factors, by far and away, risk factors that are associated with a chronic inflammatory state of mucosal surfaces are closely linked to the risk of developing cardiovascular disease. And probably the most important one are the lungs and cigarette smoking that causes a chronic inflammatory state of the lungs, and along with air pollution, both of which show high risk for developing cardiovascular disease. But the other interesting one that is more relevant to what I will be discussing later in this talk is the gut, because in fact, dietary habits, and especially the so-called Western diet, which are high in fat and high in refined carbohydrates, but low in complex carbohydrates, and also have a low intake of plant foods with a diversity of phytochemicals, and especially polyphenols, carotenoids, and many others, are closely associated or have also a very high risk of atherosclerosis and cardiovascular disease. And these are also linked to dyslipidemia, or abnormal blood lipids, and hypercholesterolemia is one of the most important ones. Now, it's interesting because everybody knows about low-density lipoproteins and the so-called bad cholesterol, but what is very important to understand about low-density lipoproteins and cholesterol is that that particular lipoprotein is not atherogenic. It is the oxidized form of this particle. That is very important in terms of the propagation and development of atherosclerosis. And so hypercholesterolemia, yes, is linked to cardiovascular disease, but the cause and effect relationship, I think, is still quite unclear. And it's no doubt that low levels of high-density lipoprotein cholesterol are also associated with a higher risk of atherosclerosis. Now, this topic could take probably several seminars, so I'm not going to talk any more about this problem, but it's an important aspect of the development of the disease, and our diagnosis of the risk solely based on cholesterol, which in fact, prior to having a major cardiovascular event, is how the medical profession looks at the problem. And that has actually led to the prescription of statins to lower blood cholesterol. Anyway, the metabolic syndrome is another problem which is fascinating from the overall problem of these behavioral issues as it relates to the development of atherosclerosis. And the metabolic syndrome is also of epidemic problems, especially in developed countries, but is rapidly increasing in developing economies. And I look at this problem as overall in atherosclerosis and cardiovascular disease, a result of the Industrial Revolution and urbanization, and I might actually also add longevity, but that's a topic for future discussion too. So why cranberry polyphenols? And so I'll be talking mostly about cranberry polyphenols in the rest of this presentation. And that's because in Wisconsin here, we're the largest producer of cranberries worldwide. And cranberries contain a nice large diversity of polyphenols and especially proanthocyanidins. And the food industry then is interested in functional foods for prevention and treatment of cardiovascular disease. And these foods are foods and beverages, dietary supplements, and food ingredients, which include cranberries. And so the industry is really very interested in promoting cranberries for their health benefit. So we have been doing research on cranberries since and human health since the late 1990s. And our first experiments really was to separate the polyphenolic components of cranberries into their main fractions. And so the main phenolic constituents in cranberries are the hydroxycinnamic acids, the anthocyanins, the flavonol glycosides, and also the proanthocyanidins. And so when you try to separate a complex plant matrix into its polyphenolic constituents, it is actually quite challenging. And in my opinion, that's one of the problems that we have in studying foods and their polyphenolic composition as it relates to health. But anyway, we've been able to modify these separations on Cefidex LH20, which is a very interesting solid phase, and I'd be happy to discuss that with anybody sometimes. But we were able to obtain fractions that are relatively pure in proanthocyanidins, and actually when you do this elution, the proanthocyanidins are retained onto the LH20 until you elute with methanol and acetone. And so these last two fractions are highly enriched in proanthocyanidins and do not contain the other phenolic fractions. So our first experiment was as it relates to the oxidation of low-density lipoproteins, and we found a very fascinating response, because this is a method where you actually use copper to catalyze the oxidation of the low-density lipoproteins. And that's because you form conjugated dienes very quickly, and they absorb at something like 234 nanometers. So you can follow the oxidative process by monitoring your solution. Now, in this particular experiment, we actually isolated the serum from human subjects and then added these fractions to the serum and then isolated the LDL. And that's important because if you just added the polyphenolic fractions to the LDL after its isolation, then most all of them would have antioxidant activity. But as you could maybe realize from the previous slide, where in fact it's endocytosis or phagocytosis of the low-density lipoprotein that leads to oxidation in the phagolysosome of the macrophage, really then in order to have a biological activity, the polyphenol should actually be associated with the LDL particle, and that's what this data suggests quite strongly. And in fact, the fractions that don't contain proanthocyanidins, which are these first three curves, this is the control, and these are fractions that contain anthocyanins and mostly anthocyanins and flavanol glycosides. And then this fraction has a smaller percentage of proanthocyanidins. And the fraction that's almost pure proanthocyanidins had the greatest effect on LDL oxidation. In other words, preventing LDL oxidation over a much longer time and not actually reaching the maximum amount of oxidation. So that suggests that, in fact, the proanthocyanidins are specifically associating with the LDL and inhibiting oxidation. And so what are those? Well, cranberry proanthocyanins are interesting, and here's a structure that Yip Fu actually characterized in cranberry that he painstakingly isolated 3 different trimers, and so this is just an example of the trimer. So here we have a epicatechin unit that's linked through a 4-6 linkage. And then here we have another epicatechin unit through what is known as the A-type interflavan bond. This is referred to as a B-type interflavan bond. And so cranberries are characterized by oligomers that have at least one A-type interflavan bond, and our research using mass spectrometry suggests that they go up to 2 or 3. So what that means in terms of their bioactivity, well, we're trying to sort that out, but that's not the subject of this topic. I just wanted to show you what the cranberry proanthocyanidins consist of. So they're mostly catechin, epicatechin oligomers with 1 or more A-type interflavan bonds and of great dispersity and degree of polymerization. So here we have the Maldi-Toff spectra of the proanthocyanidins in cranberries from the trimer all the way up to the 16-mer in this large representation of the spectra. And then if you keep looking out at the tailing edge, you can see that there's still a pattern of increase in degree of polymerization past 16. So when you do some smoothing and look at what these masses actually correspond to, you can see we get out to a degree of polymerization of 26. Now, I don't think that's the limit, but it does suggest that indeed there's a great deal of structural heterogeneity and degree of polymerization in the cranberry proanthocyanidins. And I don't think that's unique to cranberries. I think most food plants that produce proanthocyanidins have that. Now, that is just the tip of the iceberg in terms of the structural heterogeneity of proanthocyanidins. And Veronique developed this equation here to determine the number of possible oligomers if you only have the B-type interflavan bond and catechin and epicatechin. And so if you look at what that means, so if you have a degree of 20 degrees of polymerization of 20, you have close to 10 to the 12th number of possible oligomers because of the stereochemistry around the interflavan bond only. Now that means that we're dealing with an incredibly structurally heterogeneous molecule when we say proanthocyanidin. And I've got to say that when you look at the nutritional effects of these molecules, that is an interesting challenge to sort out. Now, you know, in Ricardo's presentation, it was very interesting because of the looking at the epigallocatechin gallate and then the dimers and the trimers shows that indeed degree of polymerization has a major effect on the bioactivity of this group of molecules. And that's because of the nature of the polyphenol protein interaction, which again, Ricardo did an elegant job of demonstrating as it relates to the causative effects on celiac disease and the antigenic protein. So indeed, the association between these molecules and proteins is quite strong, mostly because of the hydrogen bonding between the vicinal hydroxyl groups and also the, for instance, and other polar biomolecules. And in this case, we're showing a sort of a representation of hydrogen bonding to a peptide bond. So proanthocyanidin is complex with proteins quite strongly, and that can lead to a complex that is actually not precipitated, but at certain ratios then of these proanthocyanidins to proteins, they form insoluble complex and precipitate out, and that's the basis of the effects of tannins. And so one of the things that we've then have been doing then is isolating the proanthocyanidins from cranberries using the methods that were outlined earlier, and seeing how those affect both in vivo and in vitro responses as it relates to inflammatory processes. And so here's an example of research we've done with macrophage cell cultures, where we look at cyclooxygenase expression using Western blots and the expression then of inducible nitrogen oxide synthase. Both of these are extremely important proteins in an inflammatory reaction of macrophages to antigens and bacteria. And so low lipopolysaccharides from mostly from E. coli are used to stimulate the macrophage inflammatory response. So in the absence of lipopolysaccharide, you don't see any of these inflammatory processes. With no proanthocyanidins, you have a high expression, and as you increase the proanthocyanidin concentration, you get a decrease in the expression of these proteins, and therefore these the proanthocyanidins are actually quite anti-inflammatory in macrophage cell culture. And we also looked at it in a mouse model of inflammation, which is to actually administer LPS to the peritoneum in mice, and then look at the response of those two proteins in lungs. And this is a model of sepsis or septic shock. And at certain levels of LPS, the mouse will actually die from the presence of LPS in its peritoneum. So we sacrifice the mouse once it starts to show the symptoms of septic shock, and then look at the proteins in the lung tissue. And indeed, you can see again that the control has very low levels of cyclooxygenase and INOS. After treatment with LPS, those are highly elevated, and then the cranberry proanthocyanidins attenuate that response. So there's no doubt that the proanthocyanidins are indeed quite anti-inflammatory. But that actually introduces a serious problem with understanding the nutritional biochemistry of proanthocyanidins. And I'm glad Ricardo actually showed his diagrams of the response to the glial antigen in the area of celiac disease, because in fact the proanthocyanidins by and large are probably not bioavailable. And this is the study that I did in Africa when I was studying tropical forages for use in feeding ruminant livestock in Africa. And we were interested in developing new forage species. So we had a species called Sesbania sesban. And in collaboration with my agronomy partners, we actually selected different accessions to grow out in sufficient quantities to feed to sheep. And so these accessions varied in their content of proanthocyanidins, and from an accession that actually did not express proanthocyanidins to one that had very high levels of proanthocyanidins. And then we fed those, the leaves of these trees. It's actually a tropical tree, so we actually harvested the leaves and fed it to sheep at 3 levels of supplementation. And what we have here is the digestion coefficient for the acid detergent lignin. And when a digestion coefficient is negative, that means that the animal is excreting more of this food component than what it ate. And lignin is a plant compound not created in animals. And so why are we getting a negative digestion coefficient? And that means that at this highest level of the accession with the highest amount of proanthocyanidin, the animal is excreting 1.6 times the amount of lignin in its feces than it ate. So is the animal creating lignin in the digestive tract? Well, no, it's not. This is happening because the proanthocyanidins are forming insoluble complexes in the gastrointestinal tract that actually then elevate the fecal fiber, lignin, and insoluble nitrogen fractions in the feces. And this happens in simple stomach animals too, and I'm sure it happens in humans. And it highly suggests that indeed the bioavailability of proanthocyanidins is quite low. So what are the implications of that observation? Well, in fact, it suggests that in fact, any health benefit of proanthocyanidins is probably elicited by effects in the gastrointestinal tract. And that's certainly the case in ruminants. Now, in ruminants, the high levels of proanthocyanidins, and we're talking about 10% of the plant, 10 to 30 or even 40% of the plant material that can contain proanthocyanidins in the diet of certain ruminant species. And so in those plants, it is quite anti-nutritional. But at much lower levels in ruminants, it actually does protect the animal from a couple of diseases. And one of them is bloat, which is when an animal eats a material that ferments very rapidly in the rumen, but also modulates protein metabolism in a beneficial way. And so when you look at the overall effects that we understand from proanthocyanidins in terms of their effects on lipid oxidation, inflammation, immunity, and bacterial adherence in the gastrointestinal tract, then it actually suggests that even though the proanthocyanidins are not absorbed, they can actually have quite a large impact on health in non-ruminant species. And that gets me to the most interesting aspect, and that is the interaction of proanthocyanidins with the gut-associated lymphoid tissue and its implication in health and disease. And so Ricardo actually showed a nice diagram of this process, and I don't think I need to go into it too much, but in fact, there's 2 components to what is known as gut barrier function, and that's because the gut, which is lined by a single layer of epithelial cells, as shown here for enterocyte, which is the most abundant cell on the epithelium of the gastrointestinal tract. And then here we also have an M cell, which is actually a phagocytic cell that actually samples the lumen and presents it to lymphoid nodules characterized by dendritic cells, which then process antigen and other materials from the gut and elicits a mucosal immune response. And so the gut is, in fact, the most important tissue for mucosal-associated immunity, or mucosal-associated lymphoid tissue in the body. So that's an extremely important. The other one is just the physical barrier that this layer presents. And we know that there are many things that can disrupt that barrier. And in fact, Ricardo was talking about celiac disease, and it does a great job of disrupting this barrier, as he showed in his presentation. But this also associated with a Western diet, parenteral feeding, and enteroliquid diets, which also can disrupt this barrier. So we've been studying parenteral feeding and enteroliquid diets as it relates to gut barrier function. Both the immunological component and the non-immunological component. And our results are quite interesting. So in fact, when you give a mouse regular food, their intestinal villi are well formed. And you can see from these purple stains, these are goblet cells, and they're quite active. Under elemental enteral nutrition, which is the feeding of a liquid that just consists of essentially amino acids and sugar and some mineral and micronutrients, the gut epithelium is highly disrupted. And this actually leads to a susceptibility to secondary infections. And when we put PACs into that elemental enteral nutrition diet, we restored the functioning of the intestinal integrity. So in fact, when we look at the effects of cranberry proanthocyanidins in this mouse model, we showed that in fact the proanthocyanidins improved the gut mucus layer and morphology, increased the number and size of goblet cells, increased tight junction proteins, also probably positively affected the gut microbiota, and one of the bacterium that we're interested in response to proanthocyanidins is Akkermansia mucinophilia, mostly because, in fact, the increase in mucin excretion is quite large in response to feeding the proanthocyanidins, and in our research, it was actually elevated above the chow treatment. And then there was increased intestinal tissue levels of cytokines that stimulated secretion and increased luminal secretory immunoglobulin A, which is the main antibody of the mucosal-associated lymphoid tissue. So in conclusion, I'd just like to say that, in fact, the future research really on the effects of polyphenols actually should focus on gut barrier function and the gut-associated lymphoid tissue and the responses in those tissues as it relates to other health outcomes. And this is a little bit of a twist of logic because, in fact, much of the research on human health and nutrition, which is highly focused on other classes of flavonoids and polyphenols than the proanthocyanidins, are focusing on post-absorptive processes and looking at the effects of metabolites in peripheral blood. And sometimes I get a little bit upset about that because, in fact, I think they're looking at the after effect and not what's going on. Because, in fact, when you look at the role of the gut as it relates to immunity, well, if 50% of immunity actually originates in the gut. And gut dysfunction and chronic inflammatory diseases are related to diseases in the gut. And those are, in fact, colon cancer and inflammatory bowel diseases like celiac disease and also Crohn's disease. And those are fascinating because they are diseases of the gut, and the effects of polyphenols on those diseases, I think, is of absolute importance in human health and nutrition. But the other one is that, in fact, the diseases associated with chronic inflammatory diseases of other tissues, and especially cardiovascular disease, becomes extremely interesting in the standpoint of how the disease develops and the fact that it is a long-term chronic disease in humans. And that gets back then to the gut barrier function and how these food components affect that process. So thank you for listening, and I'll be happy to take any questions along with Ricardo.
So thank you for these very nice presentations, and I remind all of you that you can type any question you may have in the chat box. And I think we have already a couple of questions in the chat box. Being the first one for Ricardo. What about GLP-1, so glucagon-like peptide? What about GLP-1 levels with zonulin in celiac disease? Do you have any information on these parameters value and celiac disease?
Well, on mouse models, we didn't access these two parameters. GLP, glucagon-like protein 1 or 2, their association with celiac disease is not completely clear yet. There might be some association, but it has been shown, for instance, may have more like a therapeutical action on celiac disease because of these antioxidant, anti-inflammatory, and ionotrophic properties. Zonulin, we didn't access it either on the mouse model, either the serum or in the tissue, because the mouse model I use for these experiments were mainly there were these morphological changes triggered by gliadin and indomethacin intake, but the readouts and the changes this treatment induced at the tissue level is not completely a mirror of what happens in celiac disease in people, because it's a limitation. It is impossible to find a mouse model that completely recreates what happens on humans, and in celiac disease this is particularly critical. For instance, zonulin for sure it has already been shown that it has an effect on celiac disease. It modulates tight junctions assembly. But in the case of these mouse models, for instance, what observed is that the restructuring of the mucosal epithelial layer is based on the activation not only of macrophages, but also of Mateus metalloproteinases like 2 and 9, which are involved in this remodeling of the mucosal. At the serum level, and I imagine this measuring GLP-1 at serum or zonulin, these mice typically tend to develop an adaptive branch of celiac disease, but it has to be on a very, very long term. These experiments, they can have like EGAs or EGGs against gluten or glutaminase, but it has to be a very chronic treatment for them. And we start to lose some viability on the model itself.
Interesting.
I have here another question now for Jess. So Jess, a really interesting presentation. What was the type of cranberry used? Powder, juice? How could we translate this into nutrition practice for patients with CVD and type 2 diabetes? Thanks.
Great question. And there are a large number of cranberry products out there in the market. So if you're looking at what to use as a dietary supplement, I would suggest that you be careful, because in fact the dietary supplements industry is full of rather fraudulent practices. But anyway, yes, there are in fact many. Well, in our research, we generally extract the whole fruit and then isolate the proanthocyanidins from that. But in fact, in cranberries, the proanthocyanidins represent a relatively minor component. The main components in cranberries are, in fact, the organic acids, essentially citric acid and malic acid. And in fact, the whole fruit is not sweet, and so it's quite acidic, and so it's not actually consumed fresh. And then the proanthocyanidins represent about probably of the fresh fruit on a dry matter basis, about 4% of the total mass. And then the other the other phenolic components are probably down around 1 or 2% of the dry mass. But you remember that the fresh fruit is only 10% dry matter. And so, when you look at the amount of proanthocyanidins the fruit is supplying, it's not large. On the other hand, there are many companies that are making sort of powders that are enriched in the phenolic components. And most of those powders are going into the dietary supplements industry for treating urinary tract infections, which is a fascinating story about cranberries that I didn't talk about. And so those actually can have quite high contents of proanthocyanidins depending on the manufacturers and the ingredient suppliers. And in fact, our company is highly vested in sorting that out. And so, yeah, there are some nice cranberry supplements out there that have high contents of proanthocyanidins that are being marketed for the treatment and prevention of urinary tract infections. I hope that answers your question.
Was the question
fascinating,
from Alexandra?
fascinating problem.
From Alexandra Azari-Wynne-Jones. And on the same subject, I can move to another question by Nicole Tosi. What do you think is the biological mechanism explaining the emerging evidence of the protective role of cranberry proanthocyanidins against urinary tract infections?
Yeah, well, I think it gets back to the gut.
Yeah, okay.
And in fact, a lot of my research has been motivated first by trying to figure out how an indigestible, non-bioavailable food component would affect atherosclerosis. And that led me to understanding processes in the gut and the gut-associated lymphoid tissue. And I then started looking at urinary tract infections because that's of great importance to the cranberry industry. And the etiology or the disease process of a urinary tract infection is actually oral, gut, anal, perineum, urethra. And that's because the E. coli that causes urinary tract infections, which are uropathogenic E. coli, require a transient colonization of the gastrointestinal tract before they can colonize the urethra and the bladder. And so treating urinary tract infections in the gut is probably not so far-fetched. Does that make sense?
It may.
Well, the specificity is that the uropathogenic E. coli produce specific virulence factors called fimbriae and pili. And those are external structures on the surface of the bacteria that allow them to adhere and colonize epithelial cells. And the proanthocyanidins interfere with that process big time. And we've done experiments, both in vitro and in vivo, to demonstrate that the cranberry proanthocyanidins prevent the adherence and invasion of epithelial cells. And that's extremely important, both in the gut, in the bladder, but the problem is, again, the proanthocyanidins are not bioavailable. And so, you know, that's probably not the organ in which they're having their beneficial effect. It's more likely in the gastrointestinal tract by preventing colonization of the epithelium by the uropathogenic E. coli. Now, the other issue, and this is fascinating because E. coli is an incredibly diverse organism, and there's many different phenotypes that cause different diseases. And the uropathogenic E. coli are a type of E. coli that's referred to also as extraintestinal pathogenic E. coli. So most of the pathogenicity of E. coli that we are mostly confronted with cause diarrheal diseases. And of course, that the E. coli that causes hemorrhagic colitis is a bad one, and the food industry is very aware of it. What is even more devious, though, is the ones that cause extraintestinal diseases, because they don't cause overt disease in the gut, but they do colonize the gut and hide out. But once they're outside the gut and they get access to other tissues, they can cause serious disease. And so it's a fascinating subtype of the E. coli. And so how PACs interact with those E. coli in the gastrointestinal tract is actually a very important question.
We have another question for you, Jess, here in the chat box from Yan Tong Meng. Is the composition of proanthocyanidins unique in cranberry? Will that happen, that other fruits also have proanthocyanidins but perform different effects? Are the beneficial effects of cranberry proanthocyanidins related to their polymerization degrees?
Yeah, excellent question. Excellent question. And, you know, it gets back to the that little equation that Veronique developed and the huge heterogeneity of proanthocyanidins. So the first answer is yes. In fact, all fruits and plants have differing composition and structures of their proanthocyanidins. So in fact, often, the proanthocyanidins are probably at a class of biomolecule on the level of protein in terms of structural heterogeneity, which, if we by all means understand modern biochemistry and molecular biology, structure has everything to do with function. But with the proanthocyanidins, that is an incredibly challenging question to try to answer. In other words, what is the relationship between structure and function because of that equation? And that's just with catechin, epicatechin, and the B-type interflavan bond and the conformation around that bond. So that if you have an oligomer of 20, you can end up with a million individual compounds. And when you do chromatography on the things, guess what? There's no separation. So,
the nightmare. Yeah.
But you can see why Ricardo was using purified simple structures in his research on the mechanisms, because doing it with whole proanthocyanidins would be very challenging. Although
at the molecular level, it's interesting to have individual compounds, but of course, using fraction with a very complex matrix, it's something different and much more challenging to analyze.
Absolutely.
We have here another interesting question for you from Silvia Arzola. Has it been considered to supplement proanthocyanidins with probiotic microorganisms that have the ability to enhance their bioavailability once digested by mice? So we have here a combination of proanthocyanidins with probiotics aiming to enhance the bioavailability on the once digested by mice. What do you think about that?
Well, bioavailability is a loaded word. And so if you really look at the problem systematically, and really a lot of it does have to do with microbial metabolism. And the current sort of thinking out there is that that proanthocyanidins are metabolized by bacteria in the gastrointestinal tract to metabolites that are then bioavailable, and bioavailable in quotes. And the major target metabolite for that is this phenyl gamma valorolactone, which is a fascinating metabolite, and is actually a microbial metabolite of catechin. But a catechin is not a proanthocyanidin, and one could easily argue that the dimers are barely tannins, because they don't have the same effect as the trimers and higher degrees of polymerization in their bioactivity. And so the definition of bioavailable and what is interacting with the gut microbiota as in terms of metabolites becomes quite challenging. I'm rather skeptical of the research that suggests that the higher degrees of polymerization of the proanthocyanidins are bioavailable. And so I'm skeptical of any type of research. Now, in terms of mixtures of bacteria and probiotics, that's very interesting. And we've actually studied that in relationship to E. coli. And that's interesting because, in fact, the packs have a very specific effect on E. coli, but don't affect the lactobacilli, which are the main source of probiotics in terms of supplementation in the human diet. And so that's an interesting feature. It's mostly because of most of the lactobacilli are not colonizing and don't have a niche in the gastrointestinal tract. Actually, in fact, lactobacilli in adults are not a very important part of the microbiota. But that's another issue altogether. I better leave it at that. I'm sort of dominating the questions here.
Actually, some questions are on similar subject on the bioavailability. We have here a quite exotic question. Would you believe that it would be worth consuming 100% cranberry juice for patients with cardiovascular disease? I live in the UK and I will take a look at the available powders if you have any recommendations, I would greatly appreciate.
I can't do that actually because of my business.
Just an
Because we
example.
work across several different companies that supply ingredients and produce products. But maybe let's see. Yeah, probably contact me independent of the seminar here.
I don't know, Aude, do you see any different question in the chat box?
Yeah, we have a lot of different questions, but I do have a quick question for you, Ricardo. Why are you using mice, and did you think about using pigs? Because I was thinking that pigs might have a more like a closer gut system compared to human. And so is it just a cost issue or management issue, which I completely understand, but I think it's good if you can scale up those experiments.
No, as I mentioned, the in vivo models for celiac disease there's not so much models for it. So we have to use the models if you want to assess the effects of these kind of treatments or preventive therapies. We have to use the models that are available so far. And actually, these morphometric mice were the most close ones at the time to study these effects. Now there is another mice that can actually develop this innate branch and also the adaptive branch of celiac disease, because it was modified to super express the IL-15. IL-15 is also a cytokine that is very important for the activation of intraepithelial lymphocytes. So the science is trying to make these models more robust and more closely resembling to the celiac disease, but the pigs, I don't remember seeing any model for in pigs, so that's why I used the mice at that time.
Thank you.
Thank you.
Yeah, we have another question from Philip, who is asking, considering polyphenols, procyanidins, and tannins are almost not bioavailable, not also xenobiotic, always eaten, we are going back to anti-nutritional compounds. How can we guarantee that the tannin effect reach the gut barrier more than the nutritional bowel?
So is that directed to me, Aude?
I think so, yeah.
Okay. Good question. Yes. Well, my original research on proanthocyanidins is because they are anti-nutritional components of feeds for animal nutrition. Those are at much, much higher levels of intake than what is commonly seen in foods. And in fact, that's fascinating because I've studied many African plants that are in the diet of ruminant species, both wild and domestic. And some of those plants contain proanthocyanidins as high as 20 to 30% of their dry weight. So these are very high levels of tannins. And if you bite into a leaf that has that high of a tannin, it's extremely astringent. Yet ruminant animals eat those plants, especially the wild ruminants, such as a giraffe, when it's browsing on an acacia tree, is eating that level of proanthocyanidins in the apices and apical leaves that it consumes, and it's highly adapted to consuming those plants. So when you look at the levels of proanthocyanidins in the human diet, well, in the Western diet, it's almost non-existent unless you're a wino. And most folks recommend not drinking too much alcohol, although a little bit of red wine is certainly considered healthy. So it's really a matter of dose, and that's a fascinating aspect about most things in life. A little bit is good and too much is probably not so good. Anyway, it's a hard question to address, but I think in the human diet, really, we can consume a lot more proanthocyanidins in our diet with no anti-nutritional effect at all. There's several reasons for that. One is that our protein intake in Western diets is very, very high. And so even if we do get some complexation with protein and decrease in its bioavailability, which is important in animal nutrition, in simple stomached livestock, such as pigs and poultry, I think the cutoff point for an anti-nutritional effect is probably around 2% of the diet. And that's, at that level, there is actually a measurable decrease in the performance of the animal, but not necessarily a detrimental health effect. And that's because in growing animals or lactating animals, there's a high requirement for essential amino acids, and especially methionine and lysine. And so those are the first two amino acids that are affected by proanthocyanidins in terms of their digestion and absorption. And so it's that level that you would have to probably think about in terms of an anti-nutritional effect, but that is not going to be mediated by any overt health effect. And personally, you know, proanthocyanidins are not toxic. There's no evidence that they cause any kind of toxicity. And so they're a fascinating group of molecules.
On the same subject, you have another question here. Do you think that the giraffes experience the astringency of the acacia leaves?
Well, if they do, it doesn't bother them. They're a fascinating animal. Well, their prehensile capabilities is fascinating because they have a very large arch in the roof of their mouth. And if you watch them browse, they're actually reaching over the thorns on an acacia, and some of the thorns on acacias are like several, centimeters long, and some of them are probably close to 10 centimeters long. And the giraffe can put their mouth over the tip of a branch, pull back, and eat it. And you just go, my goodness, that's amazing. And those thorns can flatten a car tire easily.
That's how they brush their teeth. Yeah.
Exactly. Exactly.
Okay.
All right. Thank you very much. We still have probably a few questions, but we only have 3 minutes remaining. So I just want to make sure I have some time just to thank all of our speakers today. If you have a question that we didn't answer, please reach out to us. We're going to share those questions to the speakers. So thank you very much to our fantastic speakers today, Dr. Diaz, Ricardo Diaz, and Dr. Jess Reed. And also our moderator, Dr. Mateus, Nuno Mateus. Also, I would like to thank you, everyone, for your participation today. And we hope this webinar was helpful and brought some new knowledge to you. Please send us an email if you have any questions. The presentations are going to be free of charge and available on the website of the group Polyphenols, as well as on the YouTube channel of the group Polyphenol, Instagram and Twitter account. We have a lot of different options, social medias. And so before closing the webinar, I would like to remind you to please just take 2 to 5 minutes to complete the survey that you should receive in a few minutes by email. Otherwise, you can scan the QR code that you have on your screen right now. And also, I would like to mention that if you didn't register yet, I think it's not open yet, but stay tuned for ICP 2025 happening in Turku in July. So make sure you register as soon as registration is open. Thank you very much, everyone. Have a good day.
Thank you.
Thanks.
Thank you very much.
Everyone.